So I'm gonna.
Danny, you have to use the microphone.
Running up there. I know I am a very stick to the minute kind of person. I've already wasted 6 seconds, and I have a lot to talk about. So I am going to go fairly fast. If you have specific questions about things I'm saying, it's fine to interrupt me, and I'll either cut off discussion or shorten other parts of the talk. So I am done at 30 minutes. I'm going to devote most of my time to the last 3 items. I will just briefly talk about the first item here, making 4 points about bullying and peer victimization that you can see elucidated here. Point number 1, the phenomenon in question is common but understudied. Now, when we say common, it's hard to put a precise number on it because there are a range of values that very much vary with the definitions and methods that different people use to assess these constructs. I'm only going to be talking about victimization and bullying. I'm not going to talk about witnessing. And the main reason for that is the overwhelming majority of the biological literature focuses on either bullying or victimization. So that's why I'm going to talk about those constructs. Based on my own review, if I had to put a number on it, I would say about 25% of American youth can be classified at one point or another as victims at some point in their development. About 10% can be classified as bullies, and about 5% can be classified as both. So these are associated constructs. We would not expect the prevalence to be 5% if they were independent. This is one particularly good study that I'm going to be coming back to, the Great Smoky Mountain study. It's particularly important in that the quality of the methods are very strong, in that it is a randomly ascertained sample with longitudinal data with a particularly good assessment of the relevant constructs. And you can see the data here that show figures that roughly conform to the rates that I was showing here. It's impactful. I think it's impactful in many ways, bullying and victimization on mental health, physical health, and success. Probably the biggest evidence of the impact is when you look long term. You can see the consequences of bullying and victimization for many, many years. And one of your committee members has recently reviewed this literature, so I won't go over it in depth. I do have a particular interest in effects on mental health, and there is general non-specificity in most of the literature on bullying and victimization, in that it tends to be associated with many kinds of problems, but probably the best evidence of specificity is that there's a particular tie between victimization and emotional problems and being a bully and conduct problems. This shows you a data slide. Again, this is from the Great Smoky Mountain study, and this looks over about 10 years, and it looks at the impact on a bunch of different outcomes: physical health, legal behavior, wealth, social functioning in adulthood as a function of exposures, either no exposure, being a bully only, a victim only, or a bully victim. And here you can see that the bully victims fare particularly poorly. That's true in some studies, depending on the outcomes. In other studies, we'll see this for some of the biological measures, such as measures of inflammation, victims do particularly poorly. So one of the tricky things, particularly for the biological research, is that bullying and victimization is particularly complex. And I think when trying to understand biology, it's useful to look at the literature on psychopathology, because I think that the complexities in that literature mirror the complexities in biology. And one particularly helpful paper from Tom Boyce, Young-Chun Kim, and others looks at this relationship longitudinally between bullying and psychopathology. And there's 2 factors that create particular complications. One, quite common in epidemiology, confounders, the things that kind of co-segregate with bullying are associated with all kinds of bad outcomes. And then number two, there is this very complex relationship whereby bullying both precedes and follows psychiatric problems. So there's this transactional relationship between the two phenomena that complicates any attempt to understand where the chicken or the egg come from. All right, and then probably one of the most important issues that's just starting to be examined is how unique bullying is in terms of other stressors. So all different kinds of stressors, including childhood maltreatment, are known to predict bad outcomes. Probably the best paper on this issue looks at two very strong epidemiologic samples, and it compares associations in longitudinal data between various kinds of adversities with exposure to bullying. And the bottom line is, on some key measures, individuals who are exposed to bullying fare substantially worse than individuals who are exposed to other adverse outcomes. And there's a lot of questions about why this might be. We were talking at breakfast about some of these. Might it have to do with the social context of bullying, the temporal proximity to a time when mental health problems become great, or is it the fact that you're looking at a specific behavior? I guess nobody really knows, but it is a particularly important finding when you start thinking about biology. All right, so I'm going to run through three areas of biology fairly quickly. I'm going to start with genetics. And most studies of genetics in the realm of behavior start with twin studies. So this shows you findings from the best twin study on bullying. It's the E-RISK study, epidemiologic-based twin cohort that started in early childhood. Robert Plomin and others did the sampling. Main finding to take away here is that there is relatively strong heritability. You can see this refers to about 3/4 of variability in victimization is heritable, and there's also relatively strong heritability for bullying. All right, so both constructs are significantly and meaningfully, though not only mediated by genetics. Not all studies find this. Again, I think this is the best study, and for your report, it's the one that I would scrutinize most closely. There are also both parallels and some differences relative to studies of psychopathology. So research on psychopathology has often found that, quote, environmental measures like victimization, right, that's an environmental measure. Research on many kinds of environmental measures finds that they are heritable. Right, and there's a rich literature from Dr. Plomin and others describing how that can happen, and we think it relates both to what's called gene-environment correlations, so that certain kinds of environmental variables tend to co-aggregate with individual variables, and then also something called gene-environment interaction, meaning that the behavior of an individual tends to elicit or evoke certain kinds of environmental events. And one could imagine how that might operate here. This is not too surprising. So also bullying is fairly substantially heritable. When we look at other outward behaviors like attention deficit hyperactivity disorder, these estimates are comparable. And there's some thought, and there's some papers that I left that suggest that these associations with psychopathology arise through effects of bullying on stress responsivity. And these are studies that look at discordant monozygotic twins who do or do not develop problems in the context of bullying. And again, we'll talk about stress in a second. As far as molecular genetics, you know, I'm not a geneticist, but I play one on TV. That's what I tend to say whenever I talk about particular genetic findings. I'm generally not a fan of candidate gene studies, so I'm not going to talk about any of that work. Probably the most promising molecular genetic work is on telomere shortening. So telomeres sit on the ends of chromosomes, and they are involved in the maintenance and repair of chromatin structure, and telomeres tend to shorten when we age. One of the major things that has emerged in recent research is stress tends to increase the pace with which telomeres shorten, and these are data from the E-RISK study, that's that same twin study that I told you about, that looks at levels of telomere shortening between age 5 and age 10. This shows you correlations between telomere length between the two time points, and as you can see, there's greater shortening in the individuals that were exposed to 2 or more episodes of bullying. Now, how do we relate such genetic findings to other aspects of the brain or other aspects of brain function? This is an area that many scientists are struggling with quite mightily, and the NIMH has put forth a framework to look at these kinds of ideas. It's called RDoC, which stands for Research Domains of Criteria Initiative. You know, I wouldn't make too big a deal of it because I don't think it's that different from the way that people have been thinking about brain behavior relationships and individual differences for a long time. There's nothing that novel about it on the one hand. On the other hand, it is a public statement that provides a useful framework. And the idea behind RDoC is that there are domains of behavior that describe fairly specific events that we can study both in people and in model organisms such as rodents and non-human primates. And there are many of these different domains. The two that are relevant to bullying are what's called the negative valence domain and the cognitive systems domain. And these are the 4 events, if you will, that are studied. The response to acute threats, potential threat, loss, and cognitive control events. And the way that RDoC works is that it really begins by trying to define underlying neural circuits. And in organizing any thought about brain behavior relationships, the current idea is that's really where the research needs to start. And it's through these brain behavior relationships that genetic and environmental influences are thought to occur. In terms of the phenotypes, we kind of define these beginning with their most complex outward whole organism level types of constructs. So psychiatric symptoms would be the most complicated one we would think about, emotional symptoms, anxiety, if you will, and then we get more deeply into the organism as we move down from symptoms to behaviors that are expressed in the laboratory, cognitive abilities that might be demonstrated as part of those behaviors, hormones or autonomic factors that modulate these, as well as cytokine responses that modulate these, and then finally at the bottom, brain imaging that actually measures activity in the circuits. And so I'm going to apply some of this as we talk in the remaining 15 minutes or so about how we understand bullying behavior. The idea, again, is that this really starts with what we know about neuroscience. We start with specific behaviors where we know something about the underlying neural architecture. The issue with starting there is that there are many, many behaviors where we know a lot about the underlying neural architecture. So we constrain the behaviors that we think about based on what's clinically relevant. So I think for you, something that would be helpful for your report would be to try to define what are the relevant behaviors that you think need to be studied at an appropriate grain size. All right, let's talk about peripheral biological indicators. There are really a range of these. We can divide them into 3 basic groups: hormones, autonomic parameters, and inflammatory parameters. This shows you one relevant paper that I think I selected for a couple reasons. Number one, I was hoping that the author would be here. She's, I understand, stuck in Canada. But this is work from one of your committee members. The second reason I selected it is it does make the point that these relationships are particularly complex. So this paper looks at the relationship between bullying and the cortisol diurnal rhythm. So as you can see, both in boys and in girls, there's this diurnal rhythm that plays out across multiple days. This is 3 days, Monday, Thursday, and Saturday, where you can see the rhythm. The complexity arises from two factors. Number one, it differs between genders. I'll go over the particular difference in a second. And then number two, it differs in terms of level of bullying. So in the boys who are occasionally bullied have a high cortisol response relative to boys who are never bullied, where boys who are often bullied have a low cortisol response, right? So you see both increases and decreases. However, in girls, we only see a reduced response. Now, this is kind of typical of all the research on cortisol. It's incredibly complex. Few findings replicate, and at least for me, I don't know how much further we're going to get by pursuing it, but again, it's the kind of thing we could talk more about. Another particularly interesting finding, one of the few other findings that has been replicated, this is from the Great Smoky Mountain study, and it looks at the relationship between bullying and an inflammatory marker, C-reactive protein, and here this is one of the places where you can see pure victims do the worst in terms of having the highest elevations in C-reactive protein, and here where you see bullies, if anything, have lower inflammation than neither. People who are neither exposed. Again, there are a range of measures. I'm not gonna go over them in much detail. The findings are relatively inconsistent. Probably the most consistent finding is that there is some evidence of a blunted cortisol response in individuals that are exposed to certain kinds of chronic stress. The literature on bullying does reflect that. It gets complicated because it also turns out that certain characteristics, such as having a tendency to behave aggressively, are also associated with a blunted cortisol response. So that's one example of how this chicken and egg problem comes up. There are certain advantages to these approaches. It's easy to acquire the data. There are a lot of studies. You can collect the data in a representative sample, so those are definite advantages. There are disadvantages in that these kinds of measures, not just cortisol, but peripheral measures in general, are relatively removed from measures of brain function. So if your goal is to really understand the behavior, these are not the best measures. On the other hand, if the peripheral measure has value in its own right, then it makes more sense. So C-reactive protein, for example, that's an important measure because it tells us a lot about inflammation, and we care about inflammation because we know it's bad, all right? And so it really doesn't matter if there are complex relationships between that measure, C-reactive protein, and behavior, because we really very much value what that measure can tell us. That is not true for hormonal and autonomic measures that are thought to be windows into the brain, so to speak. Really, most current research in psychiatry and psychology that tries to go from a neuroscience standpoint uses brain imaging. So in the last 10 minutes, I'm going to walk you through the findings on brain imaging, both talking about brain structure and brain function. There are very few studies in bullying. There are many more studies in maltreatment, also in conduct disorder. I think the data in maltreatment are particularly important, both because maltreatment in general is associated with bullying, but also because the few available findings that do exist in bullying are most similar to the data in maltreatment. There's three basic kinds of approaches that people take to measures of brain structure. One are looking at particular brain regions to ask kind of narrow questions about different structural aspects of that region. The second is a whole brain kind of perusal of where findings might lie throughout the brain. And then a third, something called diffusion tensor imaging, or DTI, asks questions about connections among brain structures. So this is all structural MRI, which is non-invasive. It's probably the easiest method to use. And the fact that there are not very many findings in this area generally tells us that there are probably not strong relationships. That typically what happens in the imaging community is people have large structural data sets, and there are plenty of people who do, and they often look at findings with a whole host of different variables, and they typically don't report the findings if nothing emerges. So the file drawer problem, as we know it. This is the only study that looks at bullying and a pure measure of brain structure and bullying. This is from Marty Teicher, who's done a lot of work on maltreatment, childhood maltreatment. And this looks at among young adults who, as adolescents, had no history of child maltreatment, but had been exposed to verbal abuse from peers. This is a measure of DTI, and the main finding in this posterior portion of the corpus callosum, he finds a reduction in structural integrity, and this is comparable to what's found in kids with maltreatment. So again, it suggests that we can think about relationships, at least with brain structure, on the same spectrum of data and other stressors. The problem is this literature is pretty much a mess. It's sort of like the cortisol literature. I don't know that if you had to compare one literature as being more or less consistent, you'd probably flip a coin. There are as many replicated findings as there are failures to replicate. And the data really for any behavior for structural imaging are really not that strong, particularly for one-shot cross-sectional associations. So, if you really want to understand the biology of a complex behavior, it's not clear how much bang you get for structural MRI. All right, brain function has much more promise. There are three basic sets of measures. Event-related potentials. There are no studies on bullying and event-related potentials, so I'm not going to talk about those. That's probably the easiest to use technique, although it's far from easy. And then there are two other measures that I'll talk a fair amount about. Resting state functional MRI. So this is kind of one step up from structural MRI. It's almost as easy to use as structural MRI, and it gives you a functional indicator, so it's thought to be more sensitive, but it also has a whole host of problems. And then I'll talk in the end, I'll talk about functional MRI, which is the main technique that I use, that in some ways is the best technique. In other ways, it's also the most complicated. So, and we'll talk about some of the issues that arise related to those complications. All right, so this is resting state functional MRI. Basically what happens here is you put an individual in a scanner and you just tell them to rest. And then you measure connections among brain regions functionally based on time-related fluctuations in brain activity. So, the way, I'm not a huge fan of this either, by the way. Johanna and Andrea, who work with me, are probably snickering that, there are a lot of problems with it, and what does it really mean when you tell somebody to rest? It's hard to know what an individual is really doing. On the other hand, it has a lot of advantages in that it is functional, and it has been related to a lot of dynamic processes. And moreover, because it's relatively easy to use, we can use it in non-human primates and rodents. And so this shows you one study that I worked on with Ned Kalin and others, where we looked at patterns of resting state functional connectivity between the amygdala, it's a very, very important brain region, and prefrontal connectivity. One particularly interesting region is this region here, called the subgenual anterior cingulate. Childhood trauma has also been related to disruptions in functional connectivity in this region. The main limitation here is what we call the parameter space. That's the term we use in imaging. is very large. And what that means is there's almost an infinite number of things that you can tweak, both in terms of how you acquire the data and how you massage the data. And so that leads to a lot of failures to replicate. And that's kind of the big problem in this field in general. There are far more failures to replicate, and there's not much emphasis on replication relative to things like cortisol, where there's a huge interest in that. So again, I use task-based fMRI. This is the last thing I'm going to talk about. The nice thing about task-based MRI is we can think of particular behaviors and we can relate them to engagement of the brain. So I won't go over the details of this conflict task, but this looks at regulatory brain regions, including the amygdala. And here in this study, it relates regulatory functions engaged by these incongruent stimuli, for example, an afraid face with a happy word over it, and finds differences in brain function related to trauma. This is the one, and again, I left this paper with you guys, this is the one functional MRI study that looks at peer victimization. This looks at a reward task, so this relates to that response to loss construct that we talked a little bit about, and it finds this inverse relationship between engagement of this prefrontal region and level of peer victimization. I think more than anything, it's kind of an example of what you can find. I wouldn't make too much of an issue out of it, and it's for the reason that I'll show you in this next slide. So this is the kind of thing that my group tends to do, and this is probably the kind of thing that's most relevant for bullying research, is we engage a specific behavior that we think is relevant to the problem that we're studying. So this is a very famous paradigm called the cyberball task, and it's a deception task. You tell the child that they're going to be part of a ball-playing game, that there are two other kids that are hooked up to other computers all around the world. We do this kind of thing, and these two kids play catch, and at a certain point, they start excluding the other child. So you can see, just from the standpoint of ecological validity and thinking about a child with bullying, this has a lot of ecological validity, in that it mirrors the kind of psychological experience that you would think would be hard on a child. And for that reason, there's been a lot of interest in this and other paradigms. My group's, Johanna actually might, in the question and answers, can tell you about it. We've developed a similar paradigm where kids actually do bully other kids in the MRI scanner. The problem with this is that there are two papers that have used this task in kids exposed to some kind of maltreatment. This is a study that's pretty relevant to you. This is a longitudinal study where a group of kids were followed in school for 5 years during middle school. This is a European study, and these were kids who in peer nominations were viewed as unlikable and were not nominated. And what you can see here, and in a second I'll describe the problem with fMRI as I describe the second study, what you can see here, as you would, quote, expect, is that when kids who are chronically rejected are excluded, we see a greater response in this prefrontal region. All right? So we say this is what we expect because we imagine that this, this region has been linked to both physical and social pain, and we think that this in some way is a neural signature of the pain that that child is experiencing. Makes a lot of sense, great story, we all love it, right? So the problem is, now we go to another stressor, right? So this is early life stress. This is exposure to domestic violence recurrently. We can debate how similar or different these two things are, but when you do studies like this, usually you think they're similar. You don't think they're different. Exact same paradigm, exact same events, exact same brain region. And what do we find here? There's a lesser activation in early life stress. And again, you read the paper, it makes total sense. This is a regulatory brain region. We fail to engage this region. So this is the problem with fMRI. You generate the opposite results. All right, and it seemingly does not present a huge problem for the people doing the research. So for that reason, I don't know that current data on fMRI are all that helpful to you, even though they're the data that I think speak most directly to the kind of questions you'd want to answer. All right, look at that. 1 second. I'm 2 seconds over now. All right, so I talked about 4 things. Nobody interrupted me. Nobody fell asleep, so that's good. Thanks a bunch. All right.